Okay. All right. Let's continue where we left off, and hopefully I can finish the HPLC lecture and maybe start spectroscopy. We'll see. Okay. Just to recap, like always, how does temperature impact resolution? Tyler?
It can help increase resolution, but if it's too hot, then they'll.
Yes. So Tyler said it will help improve resolution because you know why you will have better resolution? Viscosity of the mobile phase will be lower, so that will enhance faster movement and also kinetic energy movement and of the compound and then reaching equilibrium faster. However, you don't want it to be too high. First of all, you might damage your column. Second, if it's just too fast of a movement, you won't get separation. Everything will come out really fast. Okay, how does flow rate impact resolution? You studied for the quiz recently, huh, Abi? We should optimize it. So if we increase it, what happens? If we decrease it, what happens? Okay.
If you decrease it, I think the retention time is wider.
Yes.
And then if you
Increase it.
increase it, thank you. It's usually sharper peaks.
Yes. So in general, with higher flow rate, you get equilibrium faster and movement faster, and rate of diffusion, which is longitudinal diffusion, will be lower. So, but too fast of a flow rate, again, we won't have good separation and peaks will come out together. So we really need to find the optimal flow rate for your particular analysis. And it's not the same for each compound because different compounds equilibrate differently, different types of columns have different stationary phase, so equilibration varies based on stationary phase, mobile phase, and constituents of interest. Okay, how does the packing and thickness of the stationary phase impact resolution?
I think like the thinner your stationary phase is, the higher or the better your resolution is.
Yes, so the thinner the stationary phase, or it is basically capillary. If we talk capillary column, they have very thin layer of stationary phase. So you reach equilibrium really fast, but you don't have capacity. So you cannot load a lot of samples. You don't have capacity, but you get really nice sharp peaks. So you have to decide, do you care for capacity or do you care for resolution? So then you would decide which way to go with your thickness of your stationary phase. Okay, what are the advantages of narrow diameter column? This is related. I'm just going to go with Ellery.
Thinner column gives better separation.
Again, thinner and longer column, you get better resolution, better separation. And this is also has to do with the packing of the stationary phase and how much stationary phase you have. Okay. What are the advantages of HPLC over low pressure LC? Go ahead, Drew.
Don't you get better?
We get better resolution because we have better packing, so you don't have a lot of space between particles of your stationary phase. So you don't have particles going in constituents analyzed going into multiple direction and being delayed on the column and widening. You have diffusion and you have really wide peaks. So with LC that is low pressure, you don't really have nice sharp peaks. They're mostly overtones, wide peaks. And also with HPLC, you get better reproducibility and you can collect fractions easier, and then your peaks are purer, more pure than low pressure LC, just because of the fact that they're narrow and you have less potential of co-elution. What is a gradient elution in HPLC? Gradient elution, yes, Saj?
It's like to optimize.
Yes. Yeah, everything you said is correct. It's just to simplify a little bit, gradient elution, you're just changing the concentration of the solvent over time. And this is to reduce your analysis time. If you have good separation, you can reduce your analysis time so that your compounds don't take forever to leave the column. They will leave eventually, but with increasing their concentration, you shorten the analysis time. Not necessarily increase the concentration in one way or the other. It depends on what type of HPLC you have. Reverse phase, you increase the solvent, organic solvent concentration. In normal phase, for example, you increase the water concentration. So it really depends which direction you go. What is guard column and its function? Okay, let's go with Sally.
It's like a column before the real column. Protects the real column from any.
Absolutely. So the guard column, you put it just before the column in the flow direction. And it is, the name implies, it's guarding the column, protecting it from any impurities that might bind strongly to your stationary phase and not leave the column during your analysis time, and just blocking your functional groups, reducing the capacity and efficiency of your column. Okay, how many solvents and pumps can one use in HPLC? Mike?
So when you ask that, you mean in one run or just the whole, all of HPLC?
Doesn't matter, in one run, in the entire HPLC world.
Like countless, or so many?
I mean, not counting. Yes. Yeah. Yes. Exactly, Elizabeth. So we can have one pump, but you can have multiple solvents. So you can have one solvent, two solvent, three solvent, four solvent. You can have that, and you can have a pre-pump mixing. So mixing under low pressure, and then they go pumped in one pump. Or you can have a system that has two pumps. You can, each solvent get pumped under pressure, and then the mixing comes after being pumped under pressure. You can have systems with four pumps, four individual pumps, four different solvents. But we don't usually see an HPLC run with more than four different solvents. It gets really complicated. So you can have one solvent, two solvents, three solvents, up to four solvents. All right, cool. All right, so ovens, we were talking about the different units, and I don't think we need to do ovens, but you saw that in the lab, we have a column oven, and they come in different shapes and forms, where you can have your column inside of this unit, and you regulate the temperature using that column. So for instance, you can run your HPLC without a pump. Sorry, not without a pump, without an oven. You can do that, but then there is room temperature fluctuation. So temperature fluctuation impacts your retention time. So sometimes if it's cold, then your retention time is slowed down. And then sometimes it's warm in the room, your retention time, it comes sooner. Your compound elutes sooner. So that kind of messes up with your detection. And especially if you're running standard in one day and running samples on another day, everything is going to be messed up. So even if you want to run your analysis at 23 degrees C or 24 degrees C, It's room temperature, but the room temperature is not always stable at 23. So putting it in the column and stabilizing that temperature is always a positive thing. But not every HPLC unit has a column oven. So you can function without it, but then you're under the mercy of the temperature in the room. And we talked about reducing attention time, enhancing resolution. Now, the detector, and this is where I'll spend a little bit of time talking about the different detectors. The most common detector that we use is the UV vis detector. So absorption, basically measuring absorption. That's a very common detector we use in HPLC, but not all compounds absorb light. So there are other forms of detectors that we use as well. But UV vis, I'm going to explain two different types of UV vis detectors. But again, it's based on absorption, and we'll cover absorption in the spectroscopy section of this class, but mainly chromophores. That means those compounds that absorb light, such as proteins, polyphenols. They're naturally compounds that absorb light. But there are compounds that don't naturally absorb light. We can make them absorb light by chemical reaction. So we call it derivatization, post-column or pre-column derivatization. So let's say amino acids, if we're measuring composition using reverse phase, and we have broken down our proteins into amino acids, each amino acid does not necessarily absorb light. Only 3 amino acids absorb light: tryptophan, tyrosine, and phenylalanine. Nothing else absorbs light naturally. So you add a reagent, either anhydride or OPA reagent, and then the amino acids react with this reagent and give you a compound that absorbs light. So to explain the different types of detectors, I'll go into the very common, simple detector. We call it variable wavelength detector. So in variable wavelength detector, basically you have sources of light. So the sources of light are usually a deuterium lamp, which provides light in the UV range, and then a tungsten lamp that provides light in the visible range. And in the variable wavelength detector, you have to select a wavelength. So if you remember from our lab, we talked about lambda max. So we identify lambda max, which is the wavelength at which you have maximum absorption. So we identify the lambda max for our compounds, and if we know it, then we just enter that wavelength, and the UV detector then will measure absorption at that particular wavelength using something we call as monochromator. So a monochromator, again, I'll talk about it more in detail as well, but it is kind of a unit that filters light. So you have your lamp. That doesn't work. All right, so you have your lamp, which is your source of light. So not a very good drawer. So you have your light. So it's made up of multiple wavelengths. So you get to a grating system, and the light diffracts into individual wavelengths. So you either have a grating system or a prism or both that will give you different wavelengths, lambda 1, lambda 2, whatever. You select your lambda max, and then this goes into like a slit opening that allows a very narrow bandwidth of wavelengths to go through. And let's say it's 254 nanometers, whatever it is. You select that, then this only the light at this wavelength hits your sample. Going in usually you have a flow cell where your sample goes in, and then this one particular wavelength hits the sample absorb light, and then the transmitted light gets detected and converted to absorption. So this monochromator then select that light, and then you would measure at that particular absorption at that particular wavelength. And the appropriate wavelength usually to enhance sensitivity is your lambda max. There are instances where you choose a different lambda, just because if you have interference with another compound that absorb at the same wavelength, you might go, okay, I can reduce my sensitivity and measure absorption at a different lambda. So sometimes if you have a variable wavelength detector that is a little bit advanced, it's called variable because you can select the wavelength. Sometimes you have a fixed UV vis detector, which is you can only measure at one particular wavelength. These are very old detectors. We don't get them anymore. But the variable wavelength detector, sometimes you can monitor at two channels. You can say, okay, I'm doing proteins, so I'm going to look at 214 or 220 nanometer for peptide bonds, and I'm also going to look at 280 for absorption of the phenolic amino acids. So you can select and you can have two channels. You can also program it to at different time points to detect at different wavelengths. Because then this thing moves, the slit opening moves and allows different wavelengths to pass through. But you cannot make more than two adjustments usually with such detectors. So it's limited, but it is widely used detector. Okay, yes, Brittany.
Do we go with lambda max? Usually because then it includes all the wavelengths below that. If we pick our like lambda max, that would include the wavelengths. Does that make sense?
No, I understand your question, but no, when you pick lambda max, it is a singular wavelength. So it allows to go at 254 nanometers. It doesn't allow anything below it or above it. It just allows that very narrow bandwidth of wavelength. Okay, so and lambda max doesn't necessarily mean the highest lambda that the compound can absorb. So let me draw this to explain what I meant. This is a spectra. So this is absorption. And here you have wavelength, let's say 200 to 400. So it's measuring. So here you have absorption at different wavelengths. So let's say this is 250, 50, or let's say 230. This is 254. This is 280. So this is a compound that potentially absorb at multiple wavelengths. It doesn't absorb only on one wavelength, but it so happens that its maximum absorption is at 254. It's not the highest lambda or the lowest lambda, somewhere in between. It absorbs at a range, but its highest absorption, this is lambda max. So that means higher absorption at this particular wavelength. Does that make sense now?
Thank you.
Yeah, we'll talk more about wavelength and how it works when we talk about the spectroscopy lecture. So things will fall into place. Okay. So the other one that we have used in the lab is the photodiode array detector. So the photodiode array detector functions a little differently. So here you have a photo, this you get a spectra. This here, you actually can see absorbance at different wavelengths. Okay, you don't only get a chromatogram. This is a chromatogram, this is absorption over time, okay, retention time. So this is usually your chromatogram, but this is at a particular time point, you can see absorption at different wavelengths. This is what we saw in the lab. So your caffeine, let's assume this is 16 minutes, this is your caffeine. So in the chromatogram, and we picked lambda max to be 270, but caffeine spectra is a little different, 272 it was. Then here at lambda 272 nanometer, we got a peak, we get the area, and we use that for quantification. But the PDA also gives me the spectra, the absorption spectra, which is unique for chromophores. Every chromophore has a unique absorption spectrum. So then it tells me where lambda max is and what's the range of wavelength. Here 300, if you remember, after 300, caffeine did not absorb light. So it gives me the range of lambda where caffeine absorbs light. So I can use retention time and spectra to identify that this is caffeine. So when we did our coffee sample and we saw so many peaks close to the caffeine, we used the spectra to identify which one was caffeine because they all were eluting close to each other. So the power of a PDA is it gives you a scan of the absorption over, so you get 3D data. That means wavelength scan, time, and absorption can be plotted. So you get spectra, you get chromatogram, which is very useful for not just quantification, but for identification. Now, how does the photodiode array detector work? We'll cover that in spectroscopy. Okay, so this is illustrating that when you have a regular UV detector, if you have co-elution, if you have a contaminant or a compound you're not interested in, and coming at the same retention time, you only see one peak. This other one is hidden. You don't see that there is something else. You think that's, okay, that's your compound, whatever you're looking at, fighting in this case. So you would measure the area under the curve and you go, okay, this is fine. But if I have a PDA, I'm going to see, oh, it's not really, it's multiple spectra happening there. It's not really only pyrene, there's something else in there. Then I would try to enhance my separation so that I can get a pure peak for quantification. So PDA is powerful in letting me know that there might be a contamination going on, or not contamination, co-elution.
So now when you correct for, I guess your air, will the peak stay the same after doing? And then.
Okay, so if we're able to get separation, then you'll get two peaks. And for every individual peak, one peak will just see this time. And for the other peak, we're going to see its absorption. So these will separate if we get two separation. Yeah. But if they're co-eluting, like if you remember with caffeine, in decaf coffee, we had the caffeine peak that wasn't pure. So we saw the caffeine spectra and we saw another spectra at the same time. And we said, okay, you might be overestimating your caffeine in the decaf coffee. Okay, so Any questions? Okay. Fluorescence. So sometimes your absorbing compound fluoresces in a way that you can detect fluorescence. Potentially, when what happens during absorption, and again, I'll explain that in spectroscopy. So you go from ground state to excited state. The atom or the molecule goes from ground state to excited state. So electron jumps from one shell to a higher shell. But then it doesn't stay there. It needs to relax. So it gains energy and then it needs to relax and go down to its ground state. While it's doing that, some compounds actually fluoresce. That means emit light that you can measure. And sometimes the emission is more sensitive than the absorption. So sometimes your compounds absorb light, but it's not, you don't see high peak or sensitive peak, but when they emit, the response is greater. So sometimes we need to measure fluorescence rather than absorption. But to measure absorption, your compound has to absorb. But to measure, fluorescence, your compound has to absorb light. So absorption has to happen before fluorescence can be measured. So again, fluorescence is for chromophores or for compounds that can absorb light and also fluoresce, emits light when they're going down to the ground state, from excited to ground state. So sometimes the product or the compound that you have is not a chromophore, it doesn't absorb, it doesn't fluoresce, but when you react it with a compound that will create another compound that fluoresces, then you can do the measurement. So sometimes you have a fluorescence detector that measures fluorescence at a particular wavelength that is different from the absorption wavelength. And again, I'll explain that more in spectrum. But that's the difference between the UV detector and the fluorescence. Refractive index is basically measuring the refractive index after a sample reaches the detector. So if you have water, you have, or buffer or whatever you're using, you have a very specific refractive index. If your compound is eluting, then your mobile phase now has the compound with it at the refractive index detector, then the refractive index changes, and you measure refractive index. So mostly it's used with compounds that don't necessarily absorb light, and we're not derivatizing them into anything like sugars and oligosaccharides. So these potentially use refractive index detector. So here's a question for you. In refractive index detector, can you use a gradient illusion? Think about it a little bit. What happens during gradient illusion? What are you doing? Changing the mobile phase. You cannot have, that's the limitation. When you have a refractive index detector, you cannot have a gradient illusion. You have to have only isochromatic because your refractive index changes with the change in the concentration of your mobile phase. So that will impact your detection. So another common detector is based on electrochemical reactions. So you have the pulse amperometric detector, for example, that is basically measuring oxidation reduction of analytes. So your analytes that can be oxidized or can be reduced, like phenolic compounds, for example. So you can measure change in current. So you apply a voltage and you measure change in current in a pulse amperometric detector. PAD, you know, there's PDA, photodiode array detector. This is PAD. People can confuse them, but they're both different detectors but have similar abbreviation letters, PAD versus PDA. Another type of electrochemical detector measures conductivity. So, and this is used for mostly organic acids and some carbohydrates. Another type of a detector is light scattering, and this is a universal detector because it's not really based on the chemical structure of the molecule, so it doesn't matter if they absorb light, do not absorb light. So light scattering detector is basically what happens is when your compound reaches the detector after leaving the column, the solvent evaporates, and then the non-volatiles stay and scatter light. So it's based on the scattering of the light. And the higher concentration, the more light is scattered. And that's how they link scattered light to concentration. So multi-angle laser scattering, light scattering detector, multi-angle light scattering detector is MOLS. This is a very advanced detector and often is used in size exclusion chromatography for quantification purposes. So it's just an example for an application. All right, so the other detector is mass spectrometry, mass spectrometer. So you can use that instead of, you would just link your LC. Some of you already did MS lab. So we had our regular HPLC with the reverse phase column. And instead of having a UV detector, we had the MSS detector. Chemical reactions, like I said earlier, so it's just if you have compounds that don't absorb light, you react them either before separation of the column, or sometimes you have a reactor chamber after separation. They go into reactive chamber, they react, and the compound that absorbs light is detected. Okay, any questions? No? So this is a good review or example table where if I tell you in an exam, give me an example where you would use a normal phase or reverse phase chromatography, or sometimes I give you a scenario where you want to identify mono and oligosaccharides, then you would tell me which chromatography you would use and what detection method you would use. Now, don't worry, all of these or most of them will come in later chapters. So when I cover carbohydrate, I'll give you examples of the chromatography we use and the detection method we use. This is just a summary table. So this will come up in future chapters, but this is a good summary table. We'll be talking about amino acids, vitamins, saccharides. We will have a chapter that covers all of these, which are called contaminants of concern. We'll talk about pesticides, mycotoxins, antibiotics. So now what you're studying the principle of chromatography and HPLC and GC, but later on, we're going to talk about individual components that we will utilize these methods that you're learning for the analysis. And here, just few examples, really. So application for normal phase, usually use them for hydrophilic compounds that associate with the polar stationary phase and separate for detection. Reverse phase usually is used for compounds that are known to be more hydrophobic, liking nonpolar stationary phase. And there is ion pair reverse phase. So sometimes you have species that are charged and you really want to use reverse phase. You want to enhance interaction of the column. So you block the charges on these compounds. So you use ions. You add specific components that carry charge that block charges on the molecule so that the molecule can interact via nonpolar interactions with your reverse phase. That's what ion pair reverse phase is. These are just a few examples of applying normal phase versus reverse phase. FYI, nothing here really to worry about. So ion exchange, we can use them to separate charged molecules like amino acids and proteins, but also carbohydrate, we can actually use anion and cation exchange, but we induce charge. So sugars are not charged molecules, but let's say at really high pH, they lose their hydrogen, they become negatively charged. So we're going to talk about it in the carbohydrate chapter. So you can use ion exchange for different applications. Size exclusion is usually used for purification, to just like one of the steps of purification. It's not very common in HPLC. We use it sometimes in HPLC to detect molecular weights or to separate based on size as a first dimension. And then we use it we use another chromatography following that to separate, to purify even more. So you use it to determine molecular weight, to separate as a first dimension if we're using it at high performance. But it's very common in low performance, low pressure chromatography. Affinity, so this is very specific chromatography. This is when we're trying to produce, mostly it's used for proteins, to try to separate glycoproteins, separate antigens, purify allergens, purify enzymes, or we can use it for detection of mycotoxin, which is not a protein. So these are just examples of chromatogram from ion exchange. This is size exclusion, but low pressure liquid chromatography. So you can see that the peaks are not really sharp peak. It's just a first step in purification when we want to prepare large quantities of a particular compound. So here's what we want to do, some exercise. So pull up a piece of paper, and for a +2, everybody. So on your piece of paper, you're going to answer questions that are on the next slide. And just turn it in. If you want to take a picture of your questions before you give it to me, that would be a good idea for studying, because this will not come back to you in time for your exam. Okay. So here's a chromatogram of isoflavone. Actually, I produced that when I was a postdoc. So this is a very old chromatogram, but these are standards. And I worked with this, developed a gradient, so I get good separation of at least 11 or 12, I don't remember now, isoflavones. So you want to look at this chromatogram, and here is my gradient illusion. So I start with 17% acetonitrile. Then over the first 25 minutes, I go from 17 to 25. Then over the next 5 minutes, keep it at 25. Then from 30 to 40, I increase to 30% acetonitrile. Leave it at 30% for 10 minutes. Bring it down to the original starting, 70%. In 2 minutes, and let it equilibrate the system for 8 minutes at 17%. All right, so this is ACN, stand for acetonitrile. So 83% water, 17% acetonitrile. That's my start. So with that in mind, is this normal phase or reverse phase chromatography? Sandy? Yes, it's reverse phase because your starting mobile phase is full. Even if you do not know what your stationary phase is, by just knowing that you're starting with 83% water, 17% supernitrite, and the organic solvent is increasing over time, this tells you this is a reverse phase. Okay. Which isoflavone is the most polar? Now you know it's reverse phase. So which of these? You don't have to say the name. Tell me the first one or the last one. I'm going to go with 3. The first one. So the first one is the most polar because we have the least amount of organic solvent at the beginning. So, and the stationary phase is non-polar. So it will interact the least with my stationary phase. So it will move along with the mobile phase faster than everybody else that get delayed by interacting more with the stationary phase. Okay. How would you change the mobile phase so that genistein would elute sooner without changing the relative positions of the other compounds? Okay. So what I'm asking is, how can you look from 35 potentially to 48, 47? There are no peaks. I'm asking what would you do to have genistein elute here, for example, and shorten the analysis time by 5 to 10 minutes without changing any of this? What would you do? Let's see with Madison. So you want to keep it at 25? This will take even longer because it is the least polar, right? So if you don't give it organic solvent, it won't want to come out. You want to give it, so what would you do? Madison, then instead of just staying at 30%, what I would do is go, okay, where does this compound comes out? Let's say this comes at 35. I wait a couple of minutes, and then at 40 minutes, I go up to 50%. Let's say that's 1. But then I can then bring genistein and to come here. So I don't want to do anything before 35 minutes because this is just, we interfere with all of the others. Okay. All right. So what would happen if you keep an isocratic elution mode at 17%? Page?
It will take longer for you to get the command back.
It will take longer, but what else might happen? Yeah. Say that again. We get broader peaks, and if we get broader peaks, those peaks that are close to each other will co-elute. So it becomes, you will have more diffusion, longitudinal diffusion. And because they like the stationary phase more than the mobile phase. So they want to stay longer on the cone. So your peak broadens, and when peaks broaden, they're going to overlap with each other. So not a good idea. So 2 things will happen. First, the whole analysis time will be longer. Second, it won't be efficient at all. You don't get good separation. Okay. What would happen if you keep an isocratic elution mode at 30%? So if you start with 30% acetonitrile from the beginning. Lenny? Compound will co-elute because then all of them would want to come out faster now. So then these will not separate. These are the most polar ones, will not separate. Here you will not separate. Here you will do separation. Here you will do separation. So basically, if you do that, they will all come out sooner, and then you'll get co-elution. So the other question that I don't have here is, here there is a step gradient. So what that means with step gradient is that you're not going linear from 17 to 30 right away. So you are keeping an isochromatic here in the middle. So you keep it at 25 for a little bit, you keep it at 30 for a little bit. That potentially, this isochromatic helps with separation. If I continue with also increasing linearly, I might lose separation with the peaks that are close to each other. So I make sure that I try to keep it isochromatic so that I make sure that my peaks are separated. So how do I know that? I wouldn't know right away. I start with a method and I look to see how my peaks are. I have 12 standards. If I get 5 peaks, then I have coelution. And I go back and think about my method. What should I get? I have 12 standards. I'm using standards. I should see 12 peaks. So I don't get this right away. I just work with it. And that's the fun, is when you are trying to establish a method rather than here's a method, run it. Okay, the last thing here, so turn in these papers at the end. The last thing I have in the last 3 minutes is that we often use multiple dimension separation. So if we want to purify, and especially in the protein world, if you produce, let's say, a hydrolysate, and you want to determine the most potent, the most bioactive peptide. For example, like when you break the proteins, you have peptides, and you have peptides that are antioxidants, for example. So if you want to determine which of these peptides is the most antioxidant, have the highest activity, and what is the sequence of this peptide, that could be a research question. So I'm hydrolyzing. So I start with, in order to get to that one very active peptide, I hydrolyze my protein. The first step I do, I ultrafilter. That means I remove all of the large peptides. I don't need them because I know my bioactive peptides are really small. They will be potentially less than 1 kilodalton. So I use a filter that separates everything from peptides that are less than 1 kilodalton. Then I start using several types of modes of chromatography. I can start by size exclusion, or I can start by ion exchange. Doesn't matter, either or is good. You start, let's say, by ion exchange. What you do is you get fractions. You collect each fraction and you measure activity, antioxidant activity. And you see, okay, this fraction G has the highest activity. So I collect this fraction G and subject it to size exclusion. And then I take fractions and measure the one, get the fraction that has the highest activity. And then I do reverse phase chromatography to purify it more and get the fraction that has the highest activity. And from there, I can do sequencing of the peptide to determine, to characterize that peptide. So you have multiple separation that you can do. And because, for example, peptides might have the same charge, different size. So they might have the same size, but different charge. And then you might have multiple peptides that have the same charge and the same size, but they vary in their subunit in the different amino acids, so they will react differently with the reverse phase chromatography. So multiple dimension HPLC is common for separation of compounds that have similar structural characteristics in order to get a very pure compound to identify. Okay, now you're smarter. A little bit more knowledge. Okay, we'll start spectroscopy on Friday.